Happy New Year, guys. Welcome to this year's lecture. So we will begin today with a prediction of protein interaction sites. Then next Tuesday we will talk about pair interaction prediction, and then we will get back into the variant prediction, and that essentially will conclude the semester. So remember, for this, in this context here, I talk about interactions as physical interactions, not associations, so things where you really have touch. And typically they are measured by yeast to hybrid and in a high throughput manner by crystallography. Now, when we ask how well are protein interactions inferred by homology, so by homology inference, what we're actually looking at is we have a pair of proteins A and B that interact in some organism, and we're looking at a set A prime and B prime, related proteins, sequence-related proteins, and we are asking, are these also interacting? In this context, we of course have two similarity values here, and there is now a different way in which we can use them for what I'm going to show you in the future. Essentially, the smallest one is the limiting factor, so larger than. Okay, so how well can we do that? So the first question, of course, is can we at all use similarity in different organisms? And obviously we can do that, because this is why mouse are a model system, right? If proteins in mouse would not interact, or would interact in a completely different manner from the proteins in us, then mouse could not be a very, very good model system. So how well do you believe homology inference does work? Remember, in localization it works very well, for structure it works extremely well. Then we saw that for the inference of enzyme activity it worked less well. So somehow you can say that the highest or the strongest signal comes for structure. So proteins can be 30% different at over 100 residues, still have a similar structure. For localization it's a little bit less. For enzymes it's much less. What do you believe protein interactions will be on this scale? Much less even. So let's not quite go into these numbers. What these numbers show here is the inference between worm and worm. So we're looking at A and B in worm, and A prime and B prime in worm, and depending on more similarity, so that's the HSP, the H value here, higher similarity is there, and somehow you see that it goes down, but you see also very high error bars, because when we did this analysis, we just did not have enough data. Again, so it's the distance from the HSP curve that we're looking at. Now, the next question, of course, is how does it look if we do not look at worm-worm, but I made a mistake. This is not worm-worm. This is worm versus non-worm. So these are actually what typically people would call orthologs, right? It's the corresponding pair A prime and B prime in a different organism. But in principle, we assume would work in a similar way. Now, as you see, here is what I briefly talked about. So conservation is much better for structure than this. But now let's get into a different issue. The issue of homology-based inference, essentially homology-based inference. How would it work inside of the same genome? That's sort of unexpected, isn't it?
No. I'm leaving the day after tomorrow.
Got me worried there. So the first question, I mean, homology, again, homology originally is defined as an organ that does a similar thing as a homologous organ, the finger in whale and human, for instance. Now, on the genome evolution, we are looking at different species and the different genomic makeup, and in this different genomic makeup, we would call orthologs those domains that are used in order to do the similar function. Paralogs are things that are copied, and typically we assume that they are then free, since they are copied of the same original version, they are free to do something else, because we already have the original protein in there. Now there's gene transfer. As you know, here's one example for gene transfer of a large set of incorporates entire chloroplast. There's a lot of gene transfer. Is there any other example that you are aware of for gene transfer? Jumping genes? So I want to hear why you're smiling on this one.
Because I have no idea.
Oh, okay. I thought there was some juicy story. Yeah?
This is about plasmids.
Plasmids, yes, is another example. But the first one I came across was with corn. So the different colors in corn are actually jumping genes. This is just an extreme example. So there's a lot of horizontal gene transfer. Now that implies that when we look at the genomic makeup, the set of genes, the domains that we see, then this idea that we see a clear tree is nonsense, because we constantly have jumping genetic information. So on the level of a finger, I can easily define homology, corresponding units. On the level of the genome, I cannot. I cannot define the original genome that it all started with. Why can we not do that? Why not? We could sort of sidestep the problem, right? And find the genes that are common to all life forms. And build a tree on those. Why can we not do that? Do you believe there is a subset of some genes that are common to all life forms? Yes, some proteins are, some genes are. But it is very, so when you look at some E. coli and human, and you look at what is common between E. coli and human, and possibly you look at some other bacteria, then this is not the minimal subset of what encompasses life. The Mycoplasma genitalium has 500 proteins or 500 genes, and those will not be exactly one-to-one the ones that are similar between E. coli and human. So that is not the basic set of P450 or things that are very conserved throughout evolution. They are not good sets for a genome-wide tree. They are a good set for a family of P450. That's it. That's the problem here. So essentially we have in reality to handle this gene transfer. The gene transfer makes it very difficult to say what actually is an ortholog and what is a paralog. Okay, let's get back to the issue of what I compared here is the A2', B2' given A and B, warm versus non-warm. What about inside of warm? So that's the paralog. How well would homology inference of protein interaction work? So we expect it to work much worse because we already have one copy, so we don't need to reproduce the interaction. What we observe is just the opposite. We see that the warm, the paralog, the conservation of that is much higher than the one in the ortholog. By the way, here is a similar story for a fly. This is warm, E. c. elegans fly. You see again the blue is much higher than the green. Green, blue. Blue again is the paralogs. Green is the orthologs. We have seen some, meanwhile, Predrag Radivojak has published something similar for EC numbers. For in fact Go annotations even. Why this is the case, we don't know. Clearly, there are some problems with the underlying way in which we understand the genome evolution with the cross exchange. Paralogs conserve interactions, orthologs don't, is not really true. This is an overstatement, but in terms of protein interaction, this signal is very clear. that I showed you. What does it mean in terms of using mouse models for human? Well, it means there's a problem. It doesn't mean homology-based inference doesn't work at all, but it does mean that there are more complicated issues at work here. Now, if we measured the same interaction twice, we discussed that already? So what we're going to do is we're going to have a protein pair A and B, the same two proteins, and I'm going to make another experiment observing again that interaction. My question now is the interface going to be different? For all that we know, and so we're not really looking at this example anymore, although for some of the data that I show you, we do look at similarity, but we're actually looking at identical ones here, and we look at AB, two different experiments, and we ask, are these interfaces different? Now, for the typical idea that lock key, so one protein interacts with another protein by fitting, if that idea would be 100% true, then the answer would be, clearly, these interfaces ought to be similar. Otherwise, it would not be a lock key mechanism. It would not be a fit. Now, what we show in this, and that is from Tobias Hump here, what we show here, and I'm not going to go into the details, is that this is the part where the interface is the same. This is the part where it's very, very different. So for about 10% of the protein pairs, the protein interaction, the interfaces are very different. For most of them, they are very similar. Now, you can define difference in the interface in different ways. You can say, okay, I'm asking, is it the same pairs of contacts? Or you can say, well, is it the same residues that are involved in the interface? Maybe they bind like this. It's no longer the same pairs, but it's still sort of the same surface that is buried. Or you can look at different ways of compiling similarity, and it turns out that in fact all of these capture different aspects. Here are some extreme examples. So the binding here, the green to the blue, can be like this, or it can be like the orange. That's a very different binding, right? This is clearly not lock key. Here, where we see clearly three different binding sites. This is not lock key. And a similar story is the case here. So we do see many examples where in fact protein A and B binding are observed in a different experiment, most likely because of a biological reason, binding differently. What's the reason for that? We don't know yet. But that's an important point, and that point here may have to do also with the difficulty of homology-based inference. Maybe that is sort of imprinting upon this issue of ortholog versus paralog. Now, can we predict protein-protein interactions from sequence alone? I started a project in '99. I tried it with a machine learning device that essentially looked at stretch of sequence from one protein, stretch of sequence from another protein, and it tried to predict that they are close, and it totally failed. So that got me into the odyssey of trying to reduce false positives, because that was the reason why it failed. There are simply so many things that could possibly bind. In vitro, they may. In the cell, they don't. Maybe they don't meet each other. So what I realized here is if I want to predict protein-protein interactions, then I have to begin to reduce false positives. Let me give you one example here, predict surface residues. I had developed a method that was very good at predicting the core, because I wanted to predict, in fact, residues in the inside, because I wanted to fold up proteins. Okay? Now, if you want to predict protein interactions, what you care for is the outside. So what you had to do is, in fact, you had to sort of refine that method and change it, turn it around so that it is better on the outside. Once you have that, roughly half of all the residues is sort of accessible to solvent, half is not. Now, if you now restrict yourself for protein interaction prediction to the set of things that are at the outside, you reduce by a factor of 3. So only 25%, so 1/4 of all the interactions that are possible between two proteins are getting through. Right? It's clear how this gets it to 1/4. 1/2 times 1/2, right? I'm clear so far? Okay, so better prediction of surface residues, and we have already reduced the false positives by 1/4. Okay, we're going to throw out some things. Maybe the interaction is not quite on the surface, maybe it's somewhere here and there's a pocket and I don't see that pocket from sequence, so I'm losing something. But most of what I throw out here are really false ones. So what else can we do? We could, for instance, predict residues that are involved in protein interactions. Maybe I can simply look at the sites that bind other proteins. And then maybe I can look at the sites that bind internally. That was last semester's course. Then maybe I can look at subcellular localization. And if I look at subcellular localization, you see green means more than expected, red means much less than expected. In 6 different subcellular localizations, I'm asking for a set of proteins that interact. What's the probability that they come from this, that they're both cytoplasmic? And you see that the diagonal is green. So except for extracellular proteins, most protein, you have a high enrichment of proteins that interact with each other from the same compartment. Okay? Sort of trivial. And then there are some sort of non-trivial statements in here. But that again means that if I now predict subcellular localization and I throw away everything outside of the diagonal, then again I can throw away false positives. Say my subcellular localization is sort of for these, say that those were 5 classes here and I would have a 20% in each, 1/5. Then now I'm throwing out 25 by a factor, the false positives by a factor of 25. Right? So it's 1/5, 20%, 1/5 times 1/5, 1/25. So high reduction of false positives in this. Now, when you look at the system that Tatjana and Tobias developed, where for eukaryotes we have 18 different classes, so we get from 5 classes 25, 18 times 18. So high reduction in false positives. Okay, again, I'm throwing out things that could bind, but most of the things that I throw out will not bind. Now, now we could sort of look at what is protein interaction competing with, binding other substrates. We could predict binding sites for that. And then we could predict domains, because interactions will be inside of domains. And then we would put all of this together and come up with a method that predicts protein interactions. And that essentially is a project, a research project that has taken us almost 20 years to complete. And so for the first time now since 1 year, there is a completion. So I showed this slide. I was running around for many years, showed this slide, and said, and now we put it all together and put a face, and the face has changed over the years. So finally, we have the story together, and I'll get to that on Tuesday. Now let's begin with the data collection. Let's look at the first part here. The first part being the one where we look at, can we predict the binding sites? So again, I'm looking at different interfaces here, and one question is, that's the glycoprotein 120, that's the antibody or the CD4 here. Are the residues between GP120 and the antibody or the CD4 different than the ones that you have inside of CD4? Is that the different biophysics? Okay? That's the question at hand. In particular, we look, for instance, at 3 different types of surfaces. How do we extract interactions? Where do we get them from, protein interactions? Well, by now you have done all of that. We can take PDB. There are enough structures. If we take PDB, we have to take a unique subset of the PDB, and then we get that result. For the different amino acids, we look at the surprise, to which extent the composition of that in one of the 3 classes, protein-protein, so those are 2 different proteins, internal and chain-chain. So chain-chain are 2 different parts of the same protein, okay, in blue. Have I showed this graph? Yes. I've completely gone through. How far have I gone through these plots?
Only this one. The previous slides, well, I don't remember the previous slides. Only this one.
at the lecture in enzymatic interaction.
We got into, did I get into this?
No.
So now these different colors here differ. So they all differ from 0. 0 is background. So the first thing you observe here is that binding means so two residues that bind are non-random. We cannot just connect all amino acids together and get binding, because all of these bars differ from 0. 0 is the background. Okay? That's the first observation. The second observation is that in fact these bars blue, green and red differ. The question is, are these differences meaningful? How would you answer that question?
You have to somehow calculate the significance of there are some residues like D or E where you have completely different oriented statements where the green bar goes to the up and the blue one.
So the negative charges and the positive charge, at least the arginine here, are very, very different, yes. And sometimes here are big differences in terms of numbers, but they're all in the same direction. They're all underrepresented, the sort of functional residues, the larger aromatics here. So we look at significance. Now let's just make one point clear. Meaningful, typically scientifically meaningful, means that I have an intuition for what is a difference that matters. Amino acid composition, I don't have an intuition for that. I don't have an intuition for my data set. Okay, I could add, I have to add, of course, error bars here. But this is still something beyond my scientific intuition. So, given that I don't have a scientific intuition for that, the next sort of crutch that I can use in order to answer that question is look at statistical significance. Okay? So statistical significance, how do you do that? You do, for instance, a chi-square test. The known problem with a chi-square test is when you have small data sets, it doesn't work. We have millions of data points, so that's not a problem. We see that all differences, I'm sorry, all the differences of what I showed are highly significant. The 10 minus 300, that was simply the limit of the chi-square, the machine that was used to do this chi-square in 2005. Now, the story ends and we can publish. Okay? Now, unfortunately, what happened is that Janai who did this work, Janai Ofran, had this horrible idea of looking at the statistical significance of the difference between proteins starting with A or B and proteins starting in a name with C or D. Okay? And he found that, 10 minus 300. Then he looked at proteins that were published by one author versus proteins published by two authors, 10 minus 300. And then, sort of, that was the biggest hit here. He looked at random subsets. They were slightly lower, but they clearly had an absolutely significant differences. How come?
Isn't the multiple testing problem?
No, it's not. So there are two issues. One is not very well known fact is that if you have very, very large data sets, tiny differences matter. These tiny differences could be an error in the measurement, but they immediately will be significant. Because you have a nice Gaussian, any tiny deviation for a lot of data points will deviate from the Gaussian and will therefore be very significant. Okay? So that's the sensitivity to experimental errors, or the sensitivity, or actually chi-square tests are complicated for very large data sets. That's statement number one. Statement number two, the assumption behind the chi-square is independence. That's strictly not true. Proteins are related. When you talk to a statistician, they say, well, since you cannot guarantee that these units that I have are independent, they evolved from the same common ancestor, you cannot apply a chi-square test. Unfortunately, this is sort of an explanation that is not making us very happy. That means no statistics in biology, if you pull this argument through, which is obviously nonsense. all alignment methods, whatever you are used to, apply statistics on sequences. So actually it shouldn't matter, but it does. Now, could we imagine to do statistical significance tests in a different way? What else could you test? So chi-square doesn't work, or t-tests, or whatever else we come with. The sort of traditional statistics doesn't work. Any idea how you could still test statistical significance? Data consistency. So here's an idea to test data consistency. You put, so these balls here represent the residues that interact in one of these three data sets. So there are more colors now. That is a mistake. I should have put it together from three colors. Data set 1, 2, 3. Okay. Now, what I do is I take out 1,000, or whatever number, let's begin with 1,000, from each data set. And let's take now the first one and take another 1,000 and ask these 1,000, this 1,000 that I take in the second draw, what is it most similar to? This, or this? And then I repeat that many, many, many times. Okay? And if I always match the one I took it from, then at least on a set of many, so in this particular case on a set of 1,000, I can identify the original data set. So that means there is a significant difference. If there's no significant difference, then it would randomly pick others, right? On our interfaces, this is the way it looks. I go now into 6 different types of interfaces. The one that I showed before is essentially the green one here. The real protein, the extracellular protein interactions, the transient protein interactions are here. And then there are things in between. Domain-domain is easy to understand for everybody. Homomers. The homomers are contacts between 2 identical units of the same protein. Okay? Simply a homodimer. Obligomers, they are obligatory. So the lingo means here, and that's a term we got from Janet Thornton, copied from Janet Thornton. Homo-obligomer are things where we never see that this particular chain binds anything else than itself. Homo-oligomer is the flip side of the coin. We have a homodimer, homo-oligomer, but then sometimes this is also binding something else. Okay, sometimes this binds this. Then we call it an oligomer because it has a choice. Heteroobligomer is slightly different here. What I'm talking about here is we have in proteins different units that are not really domains, but really sort of independent, more independent units than domains. They are not connected. There's a break. And they have different chains, different sequences, sorry. It's not the same. So I have this one binding to that one, okay? But it's in the same protein. So that's why it's called obligomer. You cannot away from it, get away from it. Okay, I'm born together, I could not bind. But I cannot stay away from this part because this is co-expressed, right? While these guys here, the heterooligomers, have to meet each other. Those are transient protein-protein interactions. They're on at some point of the life of the protein. Now, so we do this find self-test 1,000 times and record here how often does it find itself. And you see that the highest numbers are on the diagonal, so there's a clearly statistical difference. And when you look at this is homo, this is internal, this is somehow external, so the next level of confusion is within that class, sort of. And then when you look at the next highest level of confusion here, for instance, that is something where you have domain-domain contacts or heteroobligomer. Our heteroobligomer is 2 different chains in the same protein. This is like 2 different domains. So this confusion makes a lot of sense biologically. So we have a test that now, for this test, this will get a random result here. So if I played, and I don't have the table, all of these here, in fact, what we found out that this is the only one that did have a signal. And that turned out to be, and you guys have somehow found something in some context similar, proteins with more authors are longer. Okay? Proteins that are longer have slightly different amino acid composition. So that was a true signal. But all the others were not. And in this table, they did not show up. Okay, now we know that they differ. But the question now is, can we predict how much they differ? And in order to do this, by the way, I should have said that before, we have to define what we call a contact or proximity. Our issue here, our definition here was less than 6.5 angstrom. Then we have to do parsing of Swissprot to find what are different proteins. When we look at this feature now, and we look at in a window of 9 residues, how many of those? So protein-protein interactions could be like this, where these 3 residues that are involved in the same interface here, so that's protein A and this is protein B, these 3 are far apart in sequence. Okay? So now what I'm going to do is I'm running a window of 9, and I'm asking, in this window of 9, how many are within the interface, within this cube here, within this sphere. And there are 2 different answers here. One is when I define an interface as closest has to be 4 angstrom, yellow. Closest is 6 angstrom, blue. And you see that in both cases, so the blue is moved a little bit to the right. So the larger you make the sphere, the more residues you have in this interface. The smaller you make the sphere, you still have an average of 5 residues in 9. So that means protein interactions are mostly things that are not like this, more like that. Right? They are not like enzymatic activity. Can we machine learn how to choose the input features? How would you do that? So I give you, essentially now you have the data, you have the PDB interactions, you know what is inside a protein, you know what's outside, and you want to distinguish in a machine learning device. But what's your input feature? What are you going to use? Well, the first one is you ask somebody. Have you had a similar protein? You look at the Google, and this is what you always ought to do. Listen around. Try. It's at some point your master thesis will be about doing something new, but try to avoid duplication of things that others have done. The problem is really to find the good balance between not asking people too much and anyway, that balance is an art. In this particular case here, we used a neural network, and there are good reasons for that, in which we in fact look at, in this particular case, I show 13 consecutive residues. For these 13 consecutive residues, so that is 20 amino acids here, that's the input here, the 13 amino acids. So I try to predict with 2 output units whether or not the center in the window here is in a protein-protein interaction or not. Okay? In both of these cases, so this is prediction number 1, this is prediction number 2. In both of these cases, it's predicted to be an interaction because that unit is higher. But this is a strong prediction, this is a weak prediction. The type of units that we essentially threw at this thing is whatever we had. We, in principle, something like no, you guys haven't done that. We predict whatever we can. Secondary structure, solvent accessibility, membrane regions, disorder, aspects of function. We look at the profile and we throw it all at that. And then do some cross-validation. By the way, random split is not enough. Avoid overlap in training. Let's look at the prediction performance. And again, what I'm going to use this for here, the strength of the signal, I'm going to dial through the ROC curve. I have a performance that is higher than that. That's sort of the default threshold, if you want, for any kind of machine learning, and that gives me 1 point in the ROC curve. And then I can look only at the ones that are higher than a certain number, that increases the accuracy and reduces the coverage. And that's what I'm showing here. Coverage, so correctly predicted as opposed to observed, so the ones that I correctly predict of all the ones I could have predicted, and I'm talking about correctly predict to be an external protein interaction, and that's the accuracy of all the ones that I predicted at that threshold. Here the threshold is higher, here the threshold is lower. How many were right? If I only use sequence information, then that is this light blue curve. Now, this is the entire spectrum here. I'm going to zoom into this region here. And zooming into this region, I can show you the background random, and there are in fact different models of random. But you clearly, for a very small number of residues, you have a fairly high accuracy from sequence alone. Okay? So that is another way in which you can prove that the interfaces differ, so that the biophysics of the interface differs, because you can predict it. Right? Now, at some point here, when you increase the coverage, so when you predict more and more, when you're forced to lower the threshold, this is nowhere near the default. This is still 3% only here, 0.03%. You see that you begin to really already hit random. So sequence information gives you information. It is an important signal, but it's not getting you very far. It is giving you, on average, one residue in an interface that you can predict. Typically interfaces have about 20 residues. Okay? One you get. Don't know which one. But if you really reduce from sequence alone enough, then you have a high reliability in getting it right. Okay. What else can we use? Well, we can of course put in evolutionary information. I already said that. We can filter against this one here. And when I do all of that, this is sequence alone. This is using all the features I have, and this is random. Now, again, what I showed you before was really this tiny part of the ROC curve. Now I show the entire ROC curve here, or not the entire. I'm still only getting to 70%. I'm not sure where it hits random, but somewhere here. But you clearly see that there is a realm here where you're absolutely 10%, 20%, you're still 30%, you're still far away from random, right? Now, does that mean that the ones we predict here, the ones that we predict very well? So for some, you see that I really predict them very accurately. Have you ever heard anything that would explain why this could be the case? Have you ever heard of hotspots of protein interactions? So the idea of a hotspot in this particular interaction would be, so let me just draw out the interaction here. Well, the numbers are not right. I said that interactions have more than 20 residues. In this particular case, I would have 9. So okay, we add some points in between. Let's just assume 9. Now, a hotspot is one that is such that if I knock out this one, most of the energy of binding is gone, or some significant fraction. Okay, it could be half. At 9, 1 carries half of the energy of binding. That is a hotspot. There has been an experimental debate whether hotspots exist. So people who talked about hotspots, they said that if you plot so the energy carried by the first, by the second highest, the third highest, and the fourth, you have some exponential decline. Since you have an exponential decline, that means there are parts here that carry much more, contribute much more to the energy of binding, and those are hotspots. The people who are against that argument say, well, this is just a regular exponential, and there's nothing special in some of these. It's just a gradual decline. like the Gaussian tail. It all contributes, right? Just some contribute more than others, yes, but they are not really hotspots. They are just relatively more, okay? There's nothing specific about them. There's nothing that is recognizable. And that has been an ongoing debate. And this ongoing debate has to do with how you scale the binding energies, how you compare different experiments. There are a lot of problems underneath that led to a long-standing debate. Now, what if we could predict hotspots? Then that would lend credibility to the people who say there is something special about them. And that's exactly what we do. We simply pick out the ones that are the highest here. So when I train my system, I have residues that are involved in protein interactions and residues that are involved in ternary interactions. I never learn anything about hotspots, okay? But now I'm looking at the strongly predicted ones, and I'm calling them hotspots. It's just a word, okay? And now I'm going to see how that correlates with experimental data. This is, historically we did it here on, again, the GP120 and CD4, simply because that at the time that Jan and I did this was the one where we had most of the data for testing whether or not a residue is a hotspot. Red are the ones that experimentally tested to contribute most to the energy of binding. Okay? Purple is what we predict. Again, we never train on hotspots. We never even use the concept of hotspots. All we do is we pick the ones that are in the ROC curve that have the highest difference between the two values. Okay? Mapped to the structure, we are not using the structure, but mapped to the structure you see that there's a remarkable overlap here. Sometimes we overpredict, sometimes we underpredict, but overall this is remarkably well. Again, this only uses sequence, alignment and sequence, but it uses no information about the structure. Would that be enough to publish? Well, it's a single case, right? You need to bring more cases. When we looked at a larger set of cases, when we looked at a larger set of cases, just a sec. I apologize for that. Something wrong with my talk. So that is the method here, and that's random down here, and you see that for some set, so now this time we get a ROC curve by looking at a lot of these alanine scan measures. We did not use any of those for training. For training we used a completely different data set that had no overlap with these alanine scanning measures, and we see that some set of all of these pseudo hotspots. So for that, now we have to make a decision. When the delta G is beyond higher than some threshold, we call it hotspot. That's an arbitrary decision. And these numbers here are valid for whatever experimentalists told us should be the definition if we should take. But given that definition, there's a remarkably high overlap. So again, we are predicting delta G of binding higher than threshold without ever having trained that on the device. How could that happen? How can the system identify hotspots without having ever seen them? Any idea?
The hotspots are, I think, are so important that they may be conserved in different sequences, and so
yes.
are learned.
That is one way, this one possible explanation. And in fact, this is something, so I don't show you now, but this is not possible from sequence alone. So the evolutionary signal is a very crucial one here, which completely lends credibility to your argument. Another way in which I see that is that hotspots, they are the strongest parts of the interaction. That means they are most consistent. Yes, they are also conserved, but they're also most consistent in terms of the experimental information. So they're the ones where you find least often alternative measurements. And that's simply what the system zooms into. It zooms, the machine learning device here, picked up the most consistent signal. And those are the hotspots. Anyway, so what I find interesting here in this context is you have a prediction method that allows you to settle an experimental argument here. What is most important, this essentially is how well we predict the hotspots, and this is what I said, sequence only, hydrophobic moment only, evolutionary signal only. All of these are relatively low numbers, so it's the combination of all the information that really works at the end of the day. But this clearly here, in some sense, this jump, part of that really is only evolution. Okay, now since you can't predict the sites, you could possibly have the idea that you can use that in order to do some competitive binder kind of prediction. So here's the idea. You predict the hotspots on one molecule, you label them, and you find all the hotspots on another molecule by sequence alignments. And by this you try to predict alternative binders. That could work. And we worked on that for some time, and we had some encouraging evidence, but this project never got to a close. I believe this could still work, maybe not. Hubs. Clear what hubs are? Network hubs? So, frequent binders. All proteins have a similar number of interactions on average, or will some have more than others? Yes, we know some have more than others. So the distribution that we expect, by the way, let's get back into that. If you had never seen any data, what would you expect? So what I show is number of protein interactions per protein for all the human proteins, say, and here's an histogram. So it could be that some are like this, so this goes down, so some have more interactions, many have fewer interactions. It could be that they all have the same. It could be that there is a Gaussian. What would you expect? Well, you know the answer now. But why don't you expect a Gaussian? Why is it not a Gaussian? Whenever we see an observation, it's a Gaussian. I mean, not whenever, but in many examples it's a Gaussian. What is the thing here that makes it not a Gaussian? It looks somehow like this, you know, it's as if. What influences that?
Why should some proteins have only very few interactions? That's my problem. The left part of the Gaussian, where you would have only a really small number of proteins which have only one interaction number.
So open, I'm not sure whether I'm on the right path here, but I assume that if you looked into your car or into your bicycle, or you look into any machine and you ask yourself, how many binding situations do you have? I do believe that is essentially very, very flat. That there is a certain number of gating interactions in electric circuits that if higher, if you have more, then the circuit burns. If less, you consume too much energy. I would assume that most of the machines we build look more like that. Why? Because it's most efficient. You have an optimal operational point, and you optimize the machine or the connections in the machine for that operational point. That's what I would argue. So why is it that we don't observe that? And that ultimately comes back to this one here. When we look at settlements, or when we ask the size of cities, the size of villages or settlements of human in every single country, you see something that is not a Gaussian. And the issue here really is the additional, that's metropolitan areas in the US, on a log plot, so that is the zip right there. The important issue that you have to consider here is you have competition. And that is what you don't have in technical circuits. You have competition, meaning somehow they all start as a small settlement and then they grow and some get bigger. And that's the moment when you break the Gaussian. There is most likely, if you look at this over the human evolution, at some point when the big cities came about 10,000 years ago, maybe before that point, before you had the ability to communicate by numbers. So in order to build Ur, so Babylon, these old cities, you needed some communication on a level of written, math, and all kinds of things. Before that, you could not get to a certain size, because you simply could not administer that number of people, right? So before, things looked different, but the moment that hurdle was gone, most likely ever since there is the zip flow. So the competition part is the part, and then we expect something like the zip, something like this, and that in fact is exactly what we observe for the protein interactions. Now, since I can predict the number of interactions my protein has, I can possibly predict the binding partners or the hubness of the protein. Simply, on a micro level, I simply say more residues are predicted. On the macro level, I say I bind to more proteins. What's wrong in that logic? Can you immediately tell me where this will go wrong? So I'm going to plot, I have a method that predicts hotspots. So for every single protein in human, I'm going to predict the number of hotspots. I'm going to order them, and I'm going to see how that correlates with the number of predictions that this protein has, number of interactions that this protein has. Can you immediately see where this would go wrong? So one way in which this goes wrong is this. So I'm the same protein, I'm ubiquitin, and I bind to many other proteins, one after the other. It's one binding site, but I have different binding partners, right? And this gets us to the distinction from, oops, I'm sorry. Here we are, from Marc Vidal. Marc Vidal introduced this distinction between date and party hubs. Date hubs, have you ever heard of that? Essentially, it's really the way you imagine this. At a party, transcription factors in a protein world, that thing that I'm talking about. In a party, you stand in front of many people and you talk to them at the same time. You sort of talk to many people with different parts of your body, so to speak. Okay? While the date hub is the speed dating kind of situation. It's the same face that sits in front of a person, and then you get to the next person, next person, next person, next person. Okay? That's the date party hub idea. So the party hubs would be the ones where we have more molecular interactions. On a molecular level, I have more interactions, and then more binding partners. The date hubs, I will not see a correlation, because date hubs is just one binding site, many binding partners. Okay, let's just see what we can see. This is the two, is the same, it's just sort of fancier. The original data is here. Non-hubs, party hubs, date hubs. And you see non-hubs and date hubs are essentially the same. Here you see it better, is the gray is completely behind this purple. While party hubs look different. Okay, so we don't have a clear correlation, there is no simple signal, but I have a clear distinction. I can, from the molecular level, somehow see something that is the system level, okay? By simply predicting the number of binding sites I have. And by the way, to complete that story, so some of you may have heard of disorder. Disorder is one feature in which you sort of create different interfaces to, use the same binding site and bind different proteins by it. And these kind of interfaces, in particular the loopy ones, you see in particular in date hubs. Okay? So that is in fact the way in which you can bind with the same binding site many things, highly enriched in date hubs. That's sort of the flip side of that coin. And I was once again way, 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 way faster than I thought. Oops. I believe we have a lot of time to discuss. Or we go into... No, I'll leave it to you. So next stop will be getting a little bit more into pairs, although I briefly had that already. Just before you left. When was that? Here, right? Did I do all of that? Did we have a lecture with localization, protein interactions, and protein parent prediction and site prediction? Wow. And then I will have at least 2 lectures in which I talk about the prediction of sequence variants, the effect, how can we predict the effect of sequence variants. That brings us to the point where officially today concludes.